Hello everyone, my name is Emma Blanchette. I'm a master's student at the University of Windsor, and today I will be talking to you about the use of laser-induced breakdown spectroscopy for rapid pathogen diagnosis. Our lab group is investigating the use of laser-induced breakdown spectroscopy, or LIBS for short, to quickly detect and diagnose bacterial pathogens for use in a clinical setting. Current methods of diagnosing bacterial infections focus on determining the bacteria's phenotype or genotype and include methods such as culturing, polymerase chain reaction, and gram staining. However, these methods can take between 1 to 3 days to complete, and there is no method currently for immediate diagnosis of bacteria, which can lead to the prescription and subsequent overuse of broad-spectrum antibiotic drugs. Overuse of broad-spectrum drugs can lead to antibiotic-resistant strains of bacteria forming, making bacterial infections less treatable and patient outcomes unfavorable. Our goal then is to use LIBS to develop a rapid bacterial diagnostic technique for use in the clinical setting. Rapid characterization of the infection will reduce the use of broad-spectrum drugs, thereby lowering the rate of antibiotic resistance, and due to the speed of the technique will improve patient outcomes, lower death rates for deadly bacterial diseases, and improve clinical wait times. LIBS has enormous potential for this application because it is extremely fast. How LIBS works is a laser is focused down to the target, and the target absorbs the energy from the pulse. This causes vaporization of the target material, which generates a cloud of atoms above the surface. The cloud of atoms absorbs the remaining pulse energy, forming a microplasma. The plasma then gives off light as it cools, which is collected by our spectrometer to create a time-resolved spectrum. The entire process, from the firing of the pulse to the output of the spectrum, is accomplished in under a minute. Our setup utilizes an Nd:YAG laser circled in orange with a 10 nanosecond pulse duration and 10 Hz pulse frequency. Light is collected from the target by a steel-encased optical fiber. That is the blue fiber that's circled in dark green, and on the other end of this fiber we have an Echelle spectrometer, which takes light from the fiber and, using an intensified charge-coupled device, converts the incoming photons to a signal, resulting in a spectrum. On this slide, in the upper left corner, we show what a typical plasma looks like. This is called a laser-induced plasma, or LIP for short. As you can see from the picture, the plasma expands upwards into the air-argon environment from the surface. The resulting spectrum from shooting this piece of steel is shown in the bottom left corner, and the elements present in that spectrum are shown in the table above, highlighted in green. And the darker green indicates more of that element is present. This slide is essentially showing that the structure of the analyte is important because LIBS works best on solid, flat surfaces. These types of surfaces produce the best plasmas, and that's why we've worked very hard to develop a procedure to deposit bacteria as thin films on a substrate, as opposed to just shooting a laser into a vial of fluid. Our procedure for bacterial deposition is meant to simulate swabbing bacteria off the skin or other surfaces of the body. First, a bacterial solution is deposited onto a steel plate, which is then heated to remove excess moisture. Next, a damp swab is then used to swab bacteria off the metal plate. Then the swab is vortexed in DI water to shake off all the bacteria from the swab. Next, we show the setup of the centrifuge insert and the metal cone. A nitrocellulose filter, that little white circle, is placed on the bottom piece of the insert, the top piece is then screwed on, and then the cone is placed inside the top piece. The purpose of the centrifuge insert is to create an even layer of bacteria across that filter when it's being centrifuged, and the purpose of that cone is to concentrate the bacteria into a small place so we can detect smaller cell counts. Then the assembled piece is placed in a centrifuge tube. Then it is centrifuged. Then the filter is removed and mounted onto a steel piece with double-sided sticky tape. We took an SEM image of one of our filters, and that's shown here. And you can see that there's lawns of bacteria deposited onto the filter. Finally, we shoot the steel piece in a chamber with an argon environment, and the resulting laser shots are shown in this SEM image. So for each filter we make, we can get about 30 single shots or 30 spectra. A typical bacterial spectrum that we would see is shown here. You can see strong emission lines from magnesium, calcium, sodium, and carbon. There are also smaller emissions from phosphorus lines and argon lines to the right of the spectrum. Carbon comes from that nitrocellulose filter, so it is omnipresent in our background and we always see it. However, magnesium, calcium, sodium, and phosphorus are present in those bacterial cell membranes and cell walls, and they're present in different ratios. And taking the intensity of these ratios is how we're able to differentiate between bacteria. Argon is always present in our spectra because it's a bath gas that we use to shoot in, but it does not interfere with any of the lines that we look at, so it is fairly ideal. So how do we use these bacterial spectra? Well, we first need to be able to detect bacteria or be able to differentiate between bacterial spectra from spectra of sterile sources. And the sterile source that we use is deionized water, which we deposit using the same method that we showed on slide 5. The way that we've investigated differentiating between bacteria and water is using a multivariate technique called partial least squares discriminant analysis, or PLSDA. This technique takes user-inputted classes and independent variables, then builds a model by grouping data showing similar trends into a class. The plots generated by PLSDA are shown to the right, and the top graph depicts a discrimination between single-shot spectra. So each red point depicts a single DI water spectrum, and each green point depicts a single E. coli spectrum. The gray points on the graph show bacteria that was entered into PLSDA without any class information. So basically what we're doing here is we're giving PLSDA unclassified data, and based on the model previously built, asking it to classify each piece of data. And once it's complete, we can calculate the sensitivity and specificity to see how well it did. You can see here that for this filter, all the unclassified data was identified correctly as E. coli. The overall results for discrimination of single-shot spectra are shown in the table, and you can see that we have achieved good discrimination accuracy. However, we know that when doctors are in the clinic diagnosing someone, they won't want 30 answers, they'll just want one. So this is what motivated our next approach of summing single-shot spectra to produce one measurement per filter. The results are shown in the bottom right graph and in the table, and you can see that from this method, we get an even more reliable detection of bacteria. Now that we can detect the presence of bacteria, we actually need to be able to reliably diagnose what bacterial species is present. In past work, we investigating discriminating between genus and species, and for this task, we used discriminant function analysis, or DFA for short, because we had previously determined DFA as opposed to PLSDA to be the best algorithm for discrimination between multiple classes. That's shown in the bottom table in blue and green, in the blue and green text, and you can see that both algorithms perform very well, but DFA performs better overall than PLSDA. Our results for diagnosis between species are shown at the top right, and this discriminant function plot shows good separation between groups. This is corroborated by the number of true positives and true negatives in the table. The graph to the bottom right shows our ability to differentiate between strains, and here again we see good separation between strains and between species. These results are very promising, but they are done with a high concentration of cells, and usually in a clinical setting, one would have to deal with relatively low concentrations of bacteria, so we need to make sure that our method's limit of detection and limit of identification is as low as possible. And with that in mind, we're now trying to replicate these results with fewer cells and a non-zero background. The results here shown in the table for sensitivity are less optimistic, but the higher values of specificity are promising. As well, the figure on the right shows that there is still some visible separation between groups. We would like to increase that separation, and we're approaching that by investigating other multivariate techniques, such as artificial neural networks, to see if they perform better. We're also interested in enhancing the spectra with silver microparticles to try and help with that discrimination. And it's been shown previously that nanoparticle enhanced LIBS increases the signal-to-noise ratio, which is attributed to plasmon resonance. Our group is investigating microparticle enhancement of LIBS because they are cheaper, easier to obtain, and microparticle enhanced LIBS has never been investigated before. The silver particles we used are shown in the picture in the bottom right, and they're an average size of about 0.5 to 1 micron. An SEM image of these microparticles deposited on filter is also shown here. The spectrum shown in the slide is an example of the enhancement we see. A regular unenhanced spectrum is shown in blue, and a spectrum containing silver is shown in red. To measure the enhancement, we took ratios of line intensities of the lines that we track in bacteria with and without silver, and the average enhancement for each line is shown in the table at the top right. With silver microparticles, we see an average overall enhancement of our spectra of about 4 to 5 times larger. We're also currently investigating nanoparticle enhanced LIBS with bacteria, since no previous work has been done on this either. In the past, we've also focused on clinical specimens to see if diagnosing bacterial infections that are present in bodily fluids can be detected. To do this, a urinary tract infection was simulated and tested. We were successful in identifying the presence of Staphylococcus in urine samples and determined that the presence of urine solutes does not interfere with the classification of staph against other species of bacteria. We went one step further as well to identify the strain of staph present in urine samples, and again were successful in identifying the correct strain despite the presence of urine solutes. You can see from the figure that S. epidermidis from the urine classifies exclusively with S. epidermidis from a water sample. We're now looking to replicate these results with cerebral spinal fluid to detect meningitis infections. Meningitis is a potentially deadly disease which requires a spinal tap to diagnose, and using LIBS, we could potentially diagnose this infection in real time by shooting the CSF fluid. Applying LIBS to clinical analysis is going to be the basis of my future work. In conclusion, our group has shown that LIBS can be used to reliably detect bacteria, discriminate between bacterial species and between strains, and LIBS can also be used with good accuracy to differentiate between low-titer bacteria on a non-zero background. We've also showed that LIBS can be enhanced with silver microparticles. I'd like to thank my sponsors, NSERC and the Outstanding Scholars Research Program at the University of Windsor, and thank you for listening to my talk.